All right, it's my distinct pleasure to introduce Connor Razma, who is a BSMS student in the computational systems biology major. And so he's finishing his 5th year, and he's been working with Richard Ahn, who was part of my lab for about 4 years. And Connor, I think, was with Richard for 3, maybe?
Yeah, 3 years.
3 years. And working on a number of different clinical profiling projects that he will talk about. Connor, I think, enjoyed a lot of collaborative input, both from Richard Ahn, who in bioinformatics, but also from Elaine Reed and others, and who are more in the area of generating these clinical data sets. And so he's had a full experience around this. This actually serves a little bit like his MS honors thesis dissertation defense. And I welcome particularly Matteo Pellegrini and Aaron Maya, who are my fellow committee members. So I know that Connor will move on to a high-paying job in bioinformatics after he graduates. So our expectations are high for what he will present. Connor, take it away.
Yeah, thank you for that. And thank you everyone for being here and happy to come at a different time than normal. Because of my time restrictions, I'm gonna stay till around 12:05 and try to answer as many questions as possible. But I also just sent my email in the chat in case I go a little overtime or there's just a lot of questions. If anyone wants to email me their questions, if they don't get a chance, that's totally fine, and I can respond to them. Okay, so without further ado, I'm just gonna start presenting. And please let me know if you can't see this or if there's any other issues that anyone's having. But to start, I'm going to be presenting on baseline methylation patterns prior to flu vaccination. And without further ado, I'll begin. So just a little outline of my presentation. First, I'm going to go over some background and specifically how the data that I've been working with and how it's generated. Then I'm going to kind of combine the 2 data modalities I talk about and how I specifically got them. Then I'm going to go over the analysis as I do. A little bit of background on the analysis is not too much, but mostly the PCA that I did and also some statistical learning I did. And then I'll also give some future directions that could be done with this project, as well as just a recap of everything that we did. So first, some background on influenza. I won't go too long, but influenza is a virus that attacks the respiratory system, is known to cause coughs and sneezing. It's responsible for global epidemics, most famously the Spanish flu that many people are familiar with. It's caused by the H1N1 virus subtype, and it's not in a lot of media nowadays because of COVID, but there, in a general time, there can be up to 45 million illnesses with 800,000 hospitalizations and 60,000 deaths. So it's still a very big issue that could happen in the US or anywhere in the world. And many epidemiologists predict the next big global epidemic will also come from some sort of influenza-like virus. So it's very important that we get ahead on this and start doing work on it to prevent the next influenza and also improve our current vaccines, which is kind of what my focus is going to be on today. So a little background on influenza structure. It just has a RNA protein or virus. It's an RNA virus that has RNA in the center. This RNA is able to hijack the cell. It enters through 2 proteins called hemagglutinin and neuroamidase. These are proteins on the outside envelope that's able to use to access human cells. And they're also important for naming conventions. That's where H1N1 comes from, or H3N2. That's based off the type of hemagglutinin or neuroamidase that's on the outside. So influenza attacks the body like this. It attaches itself to a target cell. And then from there, the cell then engulfs it by accessing through those hemagglutinin proteins. From there, it makes its way to the nucleus. And at the nucleus, it releases its viral contents, including its RNA. And the RNA is able to insert itself into the nucleus. And then it from there hijacks the nucleus and starts replicating itself to produce many new viruses. And these viruses then burst out and attack the rest of the cell. And this causes the infection and causes the immune system to go into overdrive. So the human immune system composes of two parts. We have the innate immune system with dendritic cells, natural killer T cells that essentially attacks the virus and any cells in the area that the virus is infected. And this can cause a lot of the actual symptoms you have, such as a sore throat, because the cells just attack the virus indiscreetly. But that's not too much what I'm going to be focusing on today. I'm going to be focusing more on the adaptive immune system, specifically antibodies, which help track down the virus so that there can be a more targeted response to only attack the virus, as well as cells infected with the virus itself. So I'm going to be looking at that. I'm going to be looking at people's antibody response to flu vaccination. And that's kind of going to give us an idea of how well their immunity is against these different viruses. And I'll get into that in a second. So before I get into that, I just want to go over how antibodies are measured in the sample. So in our case, they were measured through the hemagglutination inhibition assay. In order to understand that, first we need to know what hemagglutination is. So hemagglutination is this process. So here, before you have any virus, say you take a bunch of red blood cells and put them in a well, there's going to be no interaction between them, and they're all going to fall down to the bottom of this well and form this larger dot. But with hemagglutination, we have these red blood cells that are combined with the virus. And this causes the virus to basically bind to the red blood cells and connect them together. And you have this plateau that sits over the well, and the well will look like this. And that's hemagglutination, and that's what's happening here. Now, what we did, or actually people at the University of Georgia who generated this data did, is they used the hemagglutin inhibition assay. So here there's the one extra step. So here's the first steps I just talked about. At the end, there's the addition of, with our actual sample, we have antibodies from our sample that are injected into this serum, and they end up binding with the virus, and they prevent the red blood cells, or they prevent the virus, from interacting with the red blood cells. And then once again, we have hemagglutin inhibition. We have this large red dot at the bottom. So for the actual hemagglutin inhibition assay, what they do is they take serial dilutions of the serum or the sample, and then they go from 110 all the way from 10 serial dilutions to 20,000 serial dilutions. And we take the highest serial dilution that still prevents hemagglutination, and we call that your HAI titer score. And that's basically just a measure of how strong your immune system is, or how, not immune system, but specifically how strong your adaptive immune system is to this virus currently. So an HAI titer score of 40 or above is considered seroprotected. That's just a way to kind of transfer the data from a count data set to a binary data set. That's just the important term. And another important thing to note about this data is it's a count-based data. So you can see it goes from 10 to 20 to 40. So it's a count structured data. There's no like in between continuous data set, like there's about 1.12 or something like that. So it's a count-based data set, and that'll be important in a second. And I also just want to mention that all that was done by the groups at UGA, the University of Georgia. Now for methylation. So DNA methylation is an epigenetic mechanism where a methyl group is added or moved to a cytosine. And this addition of a methyl group silences a base. So you can see here, this is just an example of what happened. And importantly, this silencing of a base can obviously affect your gene expression. And it's been shown in the past that often in response to stimuli, people's gene expression changes and their methylation changes as the body adapts to some sort of stimuli, or the immune system adapts. So this is another data modality we used, and we did it through reduced representation by sulfide sequencing. That's just a process here. Here's reduced representation by sulfide sequencing steps. It mostly follows normal genome sequencing, like you can see on the right, with the exception there's this bisulfite conversion. So bisulfite conversion works like this. So say you have your samples with the DNA, and there's a methyl group attached to these cytosines over here, but not here. What happens in bisulfite conversion is the cytosines that are unmethylated or converted to uracil. And then from there, you have your sequencing, and the uracil ones are sequenced as T's, and the non-cytosine or the methylated ones are still sequenced as C's. And then from there, you can study the proportion of DNA methylation at the specific site. And that's how we study DNA methylation. That's the technique to actually do it. And this was reduced representation by sulfide sequencing, which just means we did it only for a select number of sites that we actually studied by sulfide sequencing that were chosen, as we had a guess that they would be important. We didn't do the entire genome, for instance. All right, so that's a little background on all the data. Now I'm going to go into how this data kind of combines and what I'm going to be doing with it, and then the actual methods, of course, that I used in some of the results. So this entire work is part of the Collaborative Influenza Vaccine Innovation Group. So this is a multi-institutional project, CIVIC, where our goal is to create a robust influenza vaccine. So a robust influenza vaccine just means an influenza vaccine that protects against all strains of influenza for a year, whereas current influenza vaccines only protect against 4 strains of influenza. And that's why they have to do influenza forecasting. So obviously that's the goal of the whole project. This comes from the University of Georgia, as I mentioned. But obviously my goal in that project is not that large, it's much more narrow. So my goal specifically is just to find biomarkers with good vaccine response and extract these features with high importance. So basically, in this case, I'm mostly talking about biomarkers as methylated sites and see how good of a vaccine response they have. And eventually, I was also looking to get some new methylation data that is later down the line where I could see the effect of biomarkers or the effect of the vaccine on methylation data itself. I'll get more into that, but currently that data is still pending. So specifically here, I'm going to be looking at methylation effects. So I'm going to see if the methylation profile is a biomarker, essentially. If you can look at someone's methylation and say whether they have a good vaccine or a good immunity or not. And then I want to see which of these methylated sites are significant, which leads to some interpretation of what's actually going on biologically. So now to go into the data I have. So influenza obviously stems from this big word that we all talk about. Then it splits into influenza A and influenza B. And then from there, it splits into influenza A splits into H1N1 and H3N2, Yamagata and Victoria. Those are some of these subtypes that it splits into. And then all these split into historical strains. And then for the vaccine that we have one one vaccine strain for H1N1, H3N2, Yamagata, and Victoria. And that one vaccine strain is what we're going to be studying. And we're going to be studying people's immunity to that specific vaccine strain that I'm talking about right here. So the data collection was done at the University of Georgia. So the patients were given that vaccine with the 4 different influenza strains that I talked about, H1N1, H3N2, Yamagata, and Victoria. So then there was HII data taken at day 0 of vaccination. Remember that HII data is talking about someone's antibody data, essentially their immunity data. HI data was also taken at day 28 or 28 days after vaccination. So day 0 is right before vaccination, day 28 is 28 days after vaccination. So methylation data was profiled using blood samples that were also taken at the same time, day 0 or day 28. And these samples were taken 28 days after vaccination. But currently I haven't gotten any of the methylation profile samples for day 28. I only have them for day 0. And I also just want to mention that the methylation data was profiled by Elaine Weed's group and Dr. Matteo Pellegrini's group, where the vaccine data was all collected at the University of Georgia, and also the HI profiling was done at the University of Georgia as well. So now I want to give a little summary of each data modality. So for the demographic data, or just the overall data, we had 460 samples, where the data was, we had data on age, biological sex, BMI, if someone's ever smoked, and there was a mean age of 49 with a standard deviation of 17. For the HI data profile, once again, we had those 4 vaccine strains. And then for the methylation data profile, we had 13,000 methylated sites, but unfortunately we had 62 samples, so we had to cut a lot of the samples out from the original data set. But luckily, there was still a mean age of 47 with a standard deviation of 17, which is implying the overall trend of the data hasn't changed, although the significantly reduced number of samples takes a lot of the statistical power away. All right, so now let's get into some of the methods I used and then what the results looked like. The first thing I did was use principal component analysis, or PCA, to do some exploratory analysis of our methylation data and its relation to the HI data. So for those of you who are familiar, PCA is an unsupervised learning algorithm used for data reduction. It just finds the eigenvalues that retain the highest proportion of variance from the original data set. We don't need to go too much into that, but basically PCA, what is important is to know it looks like this. So say you have all these samples here on the side, 1 through N, and you have values or features 1 through K. You could then reduce this data down to principal component 1 and principal component 2, excuse me. And you can see here, this would be kind of the reduction of your data. You can see how many, what the principal component values there are. And now this allows you to do visual analysis because you only have 2 principal components or 2 components to graph on. So I use this as a data reduction tool. And then I'm going to overlay cell protection over the samples to look for any pattern. And I'll show you what that means, or what that looks like right here. So here's some results that I generated. So this is a PCA graph of that methylated sites. So those features that were reduced are the methylated sites. So all those 13,000 methylated sites were reduced down to 2 principal components. And then from there, I overlaid whether somebody was seroprotected or not seroprotected for H1N1 over here, Yamagata, and H3N2. And then over here, I overlaid their age status. So 18 to 34, 35, 49, 50, 64, and 65 plus, just to give it as a reference to the other, or just to give the age as a reference to the actual seroprotection status as well. And then the graph is just principal component 1 along principal component 2. So right away, we can see that H1N1 over here seems to follow a split along principal component 1. So over here, there seems to be people who are more seroprotected. Over here, there are people less seroprotected. If you look at principal component 1 in the overlay of the age graph, you can also see this follows somewhat of a age trend. So over here are the younger people, and over here are the older people along principal component 1. And interestingly, this follows a little bit of the literature where H1N1 seems to have a stronger immune response in people who are younger, or they have a stronger day 0 immunity to any sort of H1N1 disease. And we did not see this in Yamagata. There doesn't seem to be this kind of split. And in H3N2, unfortunately, there just doesn't seem to be enough seroprotected people to really draw that many conclusions. But this also somewhat follows the literature with influenza B strains, such as the Yamagata and also the Victoria, which I didn't show here just for space reasons. There doesn't seem to be as much trend along age that people have a different immune response. It seems to be a little more random. But here we see in the H1N1, there's a stronger day 0 immunity for H1N1. And I also want to mention that this is a little more separable. And what I mean by that is that it seems that there seems to be, like I said, more people towards one side that are not seroprotected versus people who are seroprotected. And that's going to allow us to have a little bit easier of a time with our statistical analysis that you'll see in a second, and maybe we'll have some more accurate machine learning models. Okay, so that was some of the exploratory analysis I did, a little snapshot. So now to go over the statistical learning method, so or machine learning, as it's often called. Just a second, I need some water. Okay, so I use machine learning to both to basically extract important features of these important methylated sites. So I was using this training model structure to predict immunity based off of methylation data, and I trained a model for every historical strain. So basically, I took the methylated sites, took the day 0 H1N1 or H3N2, et cetera, HII profile, and tried to predict based off of their methylated sites how well someone's HII score was going to be. And I used different models, Gaussian elastic net, which is just, for those of you familiar, that's just normal elastic net. I gave it the name Gaussian elastic net to distinguish it from Poisson elastic net, which is using a different statistical distribution, the Poisson distribution that's normally used in elastic net, which is Gaussian. And I used random forest regression, which is just a random forest regressor, and PLS regression, which stands for partially squares regression, which is basically somewhat of a similar way to using PCA and then regressing off of different principal or different components. It's basically a data reduction then followed by regression. And I used a training procedure. I used leave one out cross validation with the number of hyperparameters tuned for varying between models with 1 to 3 hyperparameters tuned for, and for each of these hyperparameters, 25 to 100 different values tried. Okay, so now I just want to show the best performing models and also where their best performance was on each vaccine strain, excuse me. So we have H1N1 over here. This is all the models and their R-squared values, how well they performed. Here's H3N2 and the best models and how they performed. As I said, we have Yamagata over here and Victoria over here down the bottom. So for H1N1, we see that Poisson elastic net performs the best. In fact, it performs the best across all of them, with the exception being Yamagata. And that kind of makes sense if you think about the data that I was talking about earlier, because remember the final dependent variable, H1N1, the final dependent variable, HII titer score, is the count data. And Poisson elastic net assumes that your dependent variable is a count data set. For those of you who are not familiar with the Poisson distribution, whereas Gaussian elastic net assumes it's a normal distribution along with the others. And we also see that the best performing model seems to be generally across H1N1, and this is just a product of it being a little more separable. H3N2 is also very accurate, but because of the nature of the close, how close those HAIR scores are together that we saw earlier, there's not really, I'd be very careful taking anything away from that. It's a little too close to really be that interesting. And then from Yamagata and Victoria, we see that the models are generally a lot less performing. And that just happens to do again with that separability. It seems to be that it's a lot harder to predict Yamagata and Victoria HAI response, at least based off of this methylation data. Okay, so now I want to go over some of the most important features, like I said, that you could extract from these. So what you would do once you take those best performing models, you can extract the most important features. And for the sake of time, I'm only showing H1N1 over here. But across H1N1, you would take the most important features. And you can see here across Poisson elastic net, Gaussian elastic net, and PLS regression, the top two most important features are the same. And it's the methylated site at chromosome 3 right here and chromosome 7 right here, where in random forest, it's a little bit different. But interestingly, this chromosome 3 region, I took, or when I did some interpretation, I looked up at the UCSC genome browser. It's a site in the exon region of a protein or gene that encodes for a transmembrane protein that's involved or has been shown to be involved in calcium influx in response to the immune system. So that kind of leads to the already kind of saying that there's probably some importance in this in the immune system response and how well your initial immunity is to any sort of H1N1 or H3N2 virus, or in this case, H1N1, excuse me, because it's already been shown to be involved in that. This chromosome 7 that also seems to be important, although albeit a lot less important than chromosome 3, was an E3 ligase, and that just means it's ligating a lot of different specific substrates, and it could have a lot of different effects on the body. And I haven't quite gone into much depth and interpretation there because there's a lot more effects it could have, but it could certainly be affecting something that's involved in this initial immune response to influenza A. Okay, so that's what I have for these that's what I have here for these most important features for H1N1. Now I want to talk a little bit about looking more into Poisson elastic net, which remember I said is the most important model, specifically for H1N1. And I want to talk about a little bit of the math behind it. And I have this math equation here for those who are kind of familiar with elastic net, but if you're not familiar with elastic net, you don't need to know the math by heart or anything like that. You don't need to completely understand this equation. I'll kind of go through the most important parts right now about it. But over here is basically the loss function for elastic net and these beta values. So here is your general loss function for just normal regression, where you're trying to minimize the loss function between your actual value and the predicted value, which is over here. And then that's divided, and this whole loss function, then you basically find the beta values through optimization that have the best performance, and that's what you take. But here with this elastic net, we do something called regularization. And regularization is basically a way to simplify the model to prevent overfitting. And that's what you do with Poisson elastic net. You add this regularizer term back here at the end. And it has a lambda value and an alpha value. And now these lambda and alpha values are very important. So the lambda values basically can control overall how much regularization you have. So a high lambda value means very high regularization and a very high amount of simplifying of the model. The alpha values, which are probably a little more important for my talk today, control which type of regularization you're going to do, whether you're going to do L2 regularization over here or L1 regularization. So an alpha value of 1 means full L2 regularization, and an alpha value of 1, means full L1 regularization, and the alpha value of 0 means full L2 regularization. That's rather important. And importantly, L1 regularization reduces a lot of your features to 0. And why that's important is that means it's literally taking features that it thinks are correlated and eliminating that correlation by eliminating one of the features completely from the model. And that's how it simplifies the model. L2 regularization doesn't do that. What it does is it basically takes all the parameters and reduces them down and makes them all smaller. But it doesn't really say that a lot of them are correlated necessarily. So you can imply that if you have a high L1 regularization compared to L2 regularization, that probably means there's a lot of feature correlation, and that a simpler model would be best by just removing a lot of the parameters. And that's kind of the overall structure, and that's kind of what you need to know, which is the large lambda value means high regularization, and a alpha value of 1 means L1 regularization. Now, looking at my best performing model for H1N1 specifically, we see that the best performing model has both a high lambda value over here and a high alpha value. So this was the best performing model that I found through that cross validation. And over here are just a couple of other examples that have a much higher, lower R squared value. So with an alpha value of 0.1 and 0.8 in these lambda values. So why this is important is now that we see that this high R squared value comes from a high alpha as well as a high lambda value, we can imply that one, there's a lot of correlation going on between our features and the methylated sites, and two, that's important that we could remove a lot of these features and we could then create a simpler model and really hone in on those most important features for interpretation that we know are going to be involved in our immune response and our innate immunity. And that's kind of the next steps I think would be in this project. So I'll just go over those future directions now and then yeah. So the next steps, like I said, just to repeat myself a little bit, is that smaller model to be more accurate and really narrow those important sites, and then really do a much more deep interpretation of those sites. And then I also want to talk about that longitudinal analysis where we can take the later methylation data and we can basically see if it's changed in the response to the vaccine. And that's really going to give insight into the immune system activation. And that's kind of another next step that can be done with that data. So then a little more farther down the road than this would be additional experiments. So once we're pretty certain on those most important sites, we can perform experiments on these sites and confirm their importance, and also maybe actually see what biologically is happening if we don't have that good of a clue of what the actual biological importance is on this immunity. And that could really help towards that final overall goal, which is by understanding the biological or methylation values, in this case, methylation sites that are really important in the immune response and really important in someone's immunity. And eventually that could help us lead to creating a stronger, more effective vaccine. And overall, that's why I discussed that we went over some strain differences. Influenza A, which is H1N1, or sorry, influenza A, my bad, has an easier ability to predict, especially H1N1 because of its separability, whereas influenza B seems a little bit harder to predict, at least through methylation. And also that influenza A seems to have more of a trend along age, where influenza B does not, which kind of follows some of the literature that if you read has been shown before. For model selection, we saw that persona-elastic net has the best performance. And finally, for feature selection, we're seeing that only a couple of these sites seem to be very important, and the whole genome is not very involved in immunity. And right before I get to questions, I just want to give some acknowledgements. First, I want to acknowledge all my thesis members who are here, Dr. Alexander Hoffmann, Matteo Pellegrini, and Aaron Meyer for going over my thesis and helping me improve it. And then I want to talk a couple other groups that have helped me. There's some overlap here. So for research mentors, this is all people who have helped me directly in my research and helped me and given me ideas as well as advice on how to conduct research and what I should do. Particularly, I want to give a thanks to Dr. Richard Ahn, as Alexander Hoffmann said, because he's been helping me a lot, and he was really my initial mentor, has been my mentor my whole time here. I also want to give a shout out for the methylation work done by Dr. Elaine Reed's group and Dr. Matteo Green's group, as well as Dr. Michael A. Carlock and Dr. Ted Ross at the University of Georgia, who did a lot of the actual setting up of the structure of how they're going to give influenza vaccines, as well as the HI data profiling. Finally, I want to thank the interdisciplinary and research program at UCLA, which Aaron Meyer is actually also a part of. And this is a group that's really helped me outside of conducting research, has helped me a lot with presenting research, presenting my own research, as well as reading new research and being able to intake different forms of research from the literature. That's also been helpful. So that's all, and I will take questions now. Thank you.